Hello, my name is Emily Kinney. I'm a second-year PhD student at the University of Toledo. I'm here to talk to you about my project today, entitled Klebsiella pneumonia, the spontaneous loss of mucoidy. But first, I have a little song to sing for you. If you know it, feel free to sing along. The itsy bitsy bacteria went up the urinary tract, in went the antibiotics to get the bacteria out, but the bacteria didn't die, instead it spread, and now you're dead. Klebsiella pneumonia, the bacteria that I study, can infect the lungs, also known as pneumonia. It can colonize the gut, which will not give you any symptoms. It can infect the blood, or bacteremia, and it can cause a urinary tract infection. In fact, Klebsiella pneumonia is the second most common cause of urinary tract infection. Additional aspects of this bacteria are: this bacteria is hospital acquired. What this means is you can get this bacteria while receiving treatment, and this is a major source of harm and death. Because you're already in the hospital getting treated for something else, your immune system's not going to be working the best. So Klebsiella is going to get you while you're down. Additionally, it is antibiotic resistant. This means that it is hard to treat. Different strains of Klebsiella can acquire little pieces of DNA that allow them to antibiotics, and antibiotic resistance spreads. This is a major source of harm and death, and this is why you should always follow the instructions when you're taking antibiotics. Two factors that I study with Klebsiella are capsule and mucoidy. The capsule is the outer sugar layer that protects the bacteria from the immune system, so it allows the bacteria to avoid being killed and recognized by cells. It says, look, I'm just a sugar, and also this capsule is thick and hard to get through. The relationship between Klebsiella and our immune system is like a war. Each side has their defenses to kill the other and survive. The bacteria has capsule, and other bacteria, not Klebsiella, can produce toxins. And then our immune system has little bacteria, and then immune cells, which this picture is inaccurate, they're actually much bigger than Klebsiella, can eat the bacteria and then kill it. Thicker capsule is more protective for the bacteria. The thinner capsule, or no capsule at all, is not good for the bacteria, but it is good for us. The immune system can more easily recognize and kill the bacteria with a thinner capsule. The other factor we study is mucoidy. When we grow Klebsiella on a plate, mucoid bacteria looks really sticky, like gum. It is hard to remove from the plate. Mucoidy requires capsule, but capsule does not require mucoidy. And what this mucoidy is the mucoidy is a less diverse capsule. So here on the left, we have a less diverse capsule chain length. These are all the same size. This means that the bacteria is less mucoid. But over here on the right, where we have all these different lengths of the capsule, this is a more diverse capsule, and that causes less mucoidy. It is associated with increased dissemination. So this means the bacteria can spread more places. So it starts off at one place of your body, say urinary tract infection, and then it can spread through your bloodstream and then to your lungs. So we don't want the bacteria to disseminate. And there may be other roles in protecting the bacteria or cause us harm. We don't know its role in the battle. We don't know if it's a shield, like capsule, or a weapon. This is one aspect of what I am researching. The other part is we took these bloodstream isolates from patients, and this one in particular had a very interesting phenotype where it lost mucoidy after it was grown multiple times. We did nothing to cause this to happen, and so now we are looking at the DNA sequence over time when the bacteria was mucoid versus when it was no longer mucoid, to see if we can determine at the DNA level why this was occurring. nucleotide polymorphisms, which are little changes in the DNA in a single base, and this can cause a big change in the phenotype because the DNA encodes the RNA, which then encodes proteins. And when these proteins aren't made correctly or have little changes, this can cause a change in the bacteria that we can then see. We also found big deletions of the bacterial chromosome, and bacteria has these circular chromosomes, which are different than what you might be thinking if you're thinking of a human chromosome. And we're not exactly sure what the loss of the mucoidy was from, because we see these SNPs. So that's my current goal, to figure out exactly why this was happening. And then, once we figure out why it's happening, can we cause this in other strains? So we have strains from other patients, and can we cause this spontaneous loss of mucoidy? And would this give us a protective effect? Additionally, we want to determine, why is this happening? And this goes back to our question on, is this a shield or a weapon? Is the bacteria purposely doing this to avoid something or attack something? Or a third option, is this just random? So, finally, to summarize, mucoidy is associated with a role in dissemination, but it might have a different part in this war between the bacteria and our immune system. And if we can control mucoidy and it has a role in the war, then maybe we can control the bacterial infection and save lives. Thank you for listening today.